So Berkshire Sterile Manufacturing was founded in 2014 by 3 individuals, all with 25 plus years of aseptic fill finish experience. And they wanted to build a brand new facility utilizing the latest innovations as it pertains to sterile manufacturing. And the way we did that was combining flex filling lines, so lines that can fill vials, syringes, and cartridges, and surrounding them with isolators for the best sterility assurance possible. Well, in March of 2016, we did our first GMP fill on behalf of a client. And at that time, isolator-based filling was brand new. And so we felt like we were bringing the industry forward by educating people on these new processes. And now the expectation here in 2022 is that people have isolator-based fill lines. The idea for the new fill line came about because we listened to our clients. And we are traditionally known to do smaller batches, and then they would have to switch to a larger CMO to accommodate their more commercial scale volumes. And so we didn't want them to have the headache of finding someone new to work with. We wanted to help them with those fills. And so we asked our team to design a line that would be able to accommodate larger batch sizes.
So we have 2 existing filling lines at Berkshire Sterile, and one is a manual, one's a semi-automated. In order to really grow and service our existing clients, we decided that we needed something more robotic in nature and that could process more product through it. And that's when we started looking at, so what would be the ideal line? How could we build this line to give our clients the flexibility that they currently have, increase the sterility assurance that we're trying to increase every time we build a line, and be there for them for the future, for future growth.
It was interesting. We held a meeting with all the sales and marketing people in the summer of 2019, and we talked to them about if we were to install a new line, what are the capacities, what are the capabilities, what are the things that you would wish in this line that would make it easier for you to go out and sell.
The business development team drafted a list of features and benefits that we wanted this line to have.
We wanted to have the flexibility to fill multiple containers. We wanted to have a continuous deparogenation tunnel for bulk containers. We wanted to have the ability to bring RTU tubs in and have them decontaminated as they came into the isolator. We wanted it to be fully automated as well, so fully robotic with glove ports, so if we needed to make an adjustment, we could do that. And we wanted to have lyophilization capability that would enable us to have much larger lot sizes for commercial production. And that's where we're like, oh, if we build this, why don't we make it so that we can add another lyo? So we have our 20,000 unit 20R vial, lyo, and in the future we can easily add on to that isolator to add another lyo of the same size. So by thinking it through and looking at how we could be flexible and how we could grow into the future, this is what we came up with.
It was certainly an, oh my God, how are we going to get all of that into a line?
We all thought it was impossible to get everything that we had wished for on that list.
Amazingly, we were able to work with 5 different suppliers and put together a really state-of-the-art isolator filling system that met all of our requirements, including being able to fill bulk and RTU vials from 2 through 100R, being able to fill and stopper cartridges and syringes. We increased the lyo capacity dramatically over our existing lyo. We can do up to 50,000 units in a single lyo run. And we also added features that really improve the sterility assurance level of this isolator versus the more traditional ones that are out there currently.
This is the greatest thing that we've ever created, and I'm going to tell you how we did it.
So So we have suppliers that we've used in the past, and we wanted to stick with EMA for the lyo, Colinar for the filler, Genesis for the capper. We actually have a new supplier, Cleanroom, for the pulse light sterilization, and of course Sterile-Line for the overall isolator construction. Once we narrowed down who the suppliers were going to be, we wanted to focus on three things with this line. That is flexibility, automation, and quality.
Well, there were a lot of challenges involving the design and creation of this new line.
There is such a wide range of components we work with, and some components that we may work with in the future, we don't even know yet.
Well, a lot of the challenges had to do with how to achieve both flexibility and speed in a sterile aseptic environment. So that included, how do you process both bulk and RTU presented containers? How do you move those containers through an aseptic environment to different machines, whether it be a lyophilizer or a capping machine? And how do you maintain that process at speed? Well, our main approach to solving some of those challenges was using what we learned through our semi-automated line to help us decide how we should design our automated line. For example, the center of our line is based around a flexible filler that's capable of filling vials, cartridges, and syringes. We also have two separate introduction pathways, one for RTU containers and one for bulk containers. And we have a separate designated diverting isolator, which is capable of sending containers either to the lyophilizer machine or directly to the capper machine. There were some processes in our semi-automated line that weren't necessarily going to be applicable to our new automatic line. For example, the depyrogenation process in our semi-automated line is a batch process.
A depyrogenation oven is a batch process. And so whenever you talk about batches, you talk about starting and stopping, sort of like a traffic jam on the highway. When you put in a continuous oven or tunnel, you eliminate that starting and stopping. So although there is a delay at the beginning of the manufacturing process, once it starts, it is continuous until it ends. And one of the natural byproducts of that is that everything that happens is consistent in the line without that starting and stopping. And so we can also call this a quality improvement as well as a speed improvement.
Another design difference included the automatic loading and unloading of some of the machines we had to use in the aseptic space, including the filling system and the lyophilization machine. In the semi-automated line, that process is entirely manual, performed by operators using glove ports. In the automated line, that had to be automated. For the filler, we had to use robotic arms to load an XY filling table with either bulk or nested containers. In the case of the lyophilization machine, we're fortunate that the vendor we selected, EMA, had a solution designed already for an automated loading and unloading system.
At this point, BSM nailed down a design that could process all container types and sizes. So, RTU containers
Goes into a no-touch transfer device where the first outer wrapping is removed, and it's then put into a pulse light decon isolator where it's hit with UV light to do surface decontamination. And then it's pushed into another isolator that has a robot that delids and delines it. And then it's carried down to the filling isolator where it's put on our filler.
and for bulk vials.
The operator brings the vials to the washer. The vials are washed and then fed into the depyrogenation tunnel. It exits the depyrogenation tunnel into another isolator, which is an accumulation isolator, which is on a turntable. And those then exit into the filling isolator, where a robot takes the vials and places them into a nested configuration and where they're filled.
How do the containers exit the line?
For syringes and cartridges, after they're filled and stoppered, they exit out of the filling isolator through wraps.
Now for liquid-filled vials,
The robot denests the vials into basically a conveyor system, which brings them into a diverting isolator, where they're then taken into the capper, a Genesis capper, and they'll be examined for raised stopper detection, as well as the crimp quality, and that's an online reject processing, and then it comes out fully capped into a collection area.
vials that are filled and need to go to lyophilizer.
That goes through the diverter isolator, and it goes down to the isolator that's in front of our lyo, and it's put onto an auto loader and loaded into the lyo. The lyophilizer is then started, and then after completion, there's an auto unload, and the vials come back out. They go back down into the diverter isolator, into the wraps Genesis capper, where they again look for raised stopper detection and quality of crimp. That gets kicked out automatically, and the vial ends up in a collection area.
So a lyo cycle can be a multi-day affair. Could a lyophil put this line out of commission?
So you can perform liquid fills while operating the lyo or doing a lyo cycle for a client. So how that works is that you would do your liquid fill and then put your vials in the lyo, and then you would break it down. The lyo is running; it's like its own separate room. You would clean it. You would VHP the areas that you needed VHP'd for your production. Run that. So, and when you bring your vials out, you would operate that as a separate fill as well, where you would set it up. You'd VHP it. You'd bring the vials out. They go into the diverting isolator and come out of the capper.
As long as that product that's on the line leaves the line to a separate space, to a separate room, then technically we can clean the line, clear it, and start again.
While a multi-day lyophilization cycle is underway, we're able to use the rest of the line to produce batches without any risk of cross-contamination.
Why are we going fully automated with this line?
Let's face it, we've all had to just come to terms with the fact that humans are not as consistent as machines are. By eliminating the operator intervention, that was a speed increase in and of itself.
We wanted automation because we wanted to be able to produce larger batch sizes for our clients' needs.
What we did was leverage robotic technology quite a bit in order to automate the line. And so what we have now is 2 robotic arms on the RTU side of the line and an additional 2 on the bulk side. So whenever we run bulk product, it sees the 2 robots in the filling chamber. And when we run RTUs, it sees all 4 robots, 2 prior to the filling chamber and then the 2 in the chamber. And then the rest is handled via conveyors and mouse holes and mouse hole doors. We can fill vials and syringes and cartridges and repeatedly with exacting standards where we could not do that with humans. The controller for the line can see where all the vials are all the time and what's going on, and in conjunction with the robots, they keep the flow going. So it's literally, we push the glass in one end, provide the product to the filler, and then wait for the material to come out the capper on the other side. It really doesn't get much more automated than that.
A major challenge was to figure out how to use the same filling system to process both bulk vials, RTU vials, and other RTU containers like syringes and cartridges, all with a single filler.
And how they did that was they relied on robots. They put 2 robotic arms in the filling chamber to replace operators, and one of them was a loading robot and the other one is an unloading robot. The loading robot would place a nest of containers onto the filling line. Now, if the nest came from an RTU bin, then that would be easy enough. But if they were working with bulk vials, then the robot would load the bulk vials into pre-sterilized nests, and then it would place that nest onto the filler. The unloading robot would remove the nest, put it back into its RTU bin, and then move it out into an exit RABS if it's a cartridge or a syringe. If the container is a vial, whether it's RTU or it's bulk, the unloading robot will take the nest off the filler, de-nest all of the vials onto a conveyor belt, and then either place the nest back into the RTU bin, if it's an RTU vial, and move those out of the filling chamber, or take the pre-sterilized nest for bulk vials and move it back towards the loading robot. So for bulk vials, they would have several pre-sterilized nests that would just circulate. And this is how they replaced operators in a filling line with just 2 robots.
Our next challenge was to overcome the possible failure modes that we would see due to the increased complexity from remaining flexible and automated at the same time.
We have raised stopper detection at 2 different locations. We have one before it goes into the lyo to make sure that the stopper is at the correct height. We also have one at the capper to make sure that the stopper's been pushed down all the way before it's capped. So we have raised stopper detection. If a vial is missing, let's say when the robot picks up the vials and loads them into the tray, if there's one missing, it can see that, and then it will pick up a single stopper and place it in that open slot. So we don't end up filling an empty slot and then putting a stopper on top of it. Some people are saying that gloveless isolators are better than gloved isolators, but we took the approach that we wanted to have operatorless isolator. It would have glove ports, so if it became necessary to save a fill, there was a problem, operators could use the glove ports to perform some operations inside. But under normal operating circumstances, the gloves would never have to be used because the robotic and the automation that we put into the system doesn't require routinely any operators to perform any tasks in the isolator. So what we again like to say is that we are operatorless, not gloveless.
By removing people, you're removing that additional risk.
The Sterile-Line automated filler has been designed with quality first and foremost in mind. This is an isolator technology. Having it be an automated line increases its sterility assurance in that we are removing humans from the system, which we don't have to rely on manipulations or anything of that nature. The robots do it for us. We also have added a new technology.
ESM was on the hunt to enhance sterility assurance for RTU tubs entering the filling line. They evaluated several different technologies, including e-beam sterilization.
But it was very large, it was expensive, and left byproducts on the surfaces of the containers.
They looked at flash steam sterilization.
But that wasn't advanced enough for our commercial use.
They also looked at sterilizing gases like VHP.
But that raised product concerns and some byproduct concerns.
Eventually, though, they did find a solution.
Pulse UV light was the best way to go. So it's safe, effective, fast, environmentally friendly, and it was cost effective. Essentially, our RTU tubs of vials, syringes, cartridges are going to go into the isolator through this chamber. Inside the chamber, there's a pulse UV light, and it's going to sanitize the surfaces of the RTU tubs as they're entering into the isolator, or right before they're entering into the isolator. Pulse light has been used in many other industries, food packaging and other things like that, and now we're adding it into our filling line for some extra sterility assurance. There is a robot inside the decontamination chamber, and it picks up the RTU tub, presents each side to the pulse UV light, and then there are several flashes that sanitize the outside of the container. And then it'll put it back down and pick it up from another position and then sanitize all of the surfaces again. And then it essentially just goes into the filling chamber from there.
With this new technology for decontamination, it's going to be completely automated. We'll be providing at least a 4-log reduction in bacterial counts as stated through our validation. In NTT, man-based versus automated, you have parameters that are validated and you can count on it every single time. You have variables when you have a human element inputted into a filling system.
Hi, my name is Stephen Kinney. I'm the Associate Director of Validation here at Berkshire Sterile Manufacturing. I'm going to give you a tour of our new fully autonomous flexible filling line. We're going to start with bulk vial introduction. Bulk vials are placed on this in-feed table here, and through a rotating table are introduced to our vial washing machine, which uses a combination of recirculated water and virgin Wi-Fi to clean the vials of all contaminants. After vial washing, vials are automatically routed to a depyrogenation tunnel, which you see in front of you here, protected by a laminar airflow unit. Inside of the depyrogenation tunnel, vials are sterilized with dry heat to achieve a 3-log reduction in endotoxin. After depyrogenation, vials are automatically fed into our filling chamber, which you see here. Bulk vials are fed in here along the left-hand side of the chamber into an in-feed scroll, where they're picked up by a loading robot and placed onto an XY table. The XY table is used by the filling system to fill and stopper vials. It's a dual-headed filling system capable of using both peristaltic and piston pumps and applying both lyo and serum stoppers. An unloading robot then takes the vials after they're filled and places them on an exit belt. Next, we'll look at how RTU vials and containers are introduced. RTU tubs are placed on this roller and handled manually by an operator using an automated blade to cut the bag and push the tubs out of the bag using a no-touch transfer method. The tubs are then loaded into a pulsed light decontamination system. The pulsed light decontamination system uses pulses of UV light to decontaminate each face and side of the RTU tubs. Inside of this chamber, which is sealed from light, a robotic arm picks up and faces each side of the tub to a set of UV lamps. After decontamination of the exterior, the tubs are brought into the delid and deliner isolator, where a robot removes the RTU lid and liner before pushing it into the filling chamber, which you've seen before. RTU tubs make their way in through the rear of the filling chamber, and the nest is picked up by the loading robot and placed directly onto the XY table. The filling process performs the same way on the XY table with vials, syringes, and cartridges in a nested format. Syringes and cartridges are picked up inside of their nest by the unloading robot and sent directly out from the filling chamber onto this unloading platform that you see to my right. Vials from both RTU and bulk lines will be placed on this exit feed belt here to be routed into our positive diverting system to either the lyophilizer or the capper. The positive diverting system is a three-way diverting system that includes lyo stopper detection and serum stopper detection and rejection. Vials are routed here either directly to the lyophilizer or to the capper. Vials will also be routed from the lyophilizer to the capper post-lyophilization. Vial capping occurs here in our Genesis RW capping system, which includes pre and post-cap vision systems for raised stopper detection and rejection, as well as crimp compliance. Completed vials are fed out onto this rotating table where they're collected in trays by the operator. The lyophilization features an automatic loading and unloading system for the loading of vials of sizes from 2R up to 30R. This sterile automated filling line is the culmination of over 3 years of work by Berkshire Sterile Manufacturing and its employees, and I'm proud today to present it to you in its entirety. This concludes the tour. I hope you enjoyed it.
I think auditors will like the innovation that is state-of-the-art equipment. It shows growth, not just for BSM as a company, but overall in the pharmaceutical industry.
Everything's unique about this new filling line. So prior to 2014, really flex filling wasn't a thing. This line offers robotic filling, fully robotic. This line offers flexibility in terms of bulk and RTU. It gives you high lyo capacity. It gives you the ability to add more lyo capacity. So there's a lot of uniqueness to this line.
Well, people are slow to adopt new technologies in this industry. I think that's one place that BSM excels, in that we take risks and we implement new technologies, and we're not following other people who have proven them out.
So I do think this is going to change the industry standard for small scale fillers. We have put in some very unique features that really improve the sterility assurance level. We've removed operators pretty much from the filling operations. We've put in a very positive decontamination step for ready-to-use containers that you typically don't see today on small volume fillers.
It took us 2 to 3 years to build this line. We built it as a team with over 5 different suppliers, and now it's ready for business.